Nature versus nurture. It's the age-old question, do our genetics or our environment have more influence on our physiology and our inherited traits? Take, for example, Billy and Bobby. Billy and Bobby are identical twins. This means they share the exact same genetic information. However, although they share a common genome, they can grow up to have very different lives, personalities, and even medical histories. For example, Billy might smoke as an adolescent and develop lung cancer, whereas Bobby's poor dietary choices might cause him to develop obesity. Now, this may not seem particularly profound, but there's actually a lot more going on at a genetic level than one might assume. Today, scientists are studying this in a relatively new field called epigenetics. Let's talk about epigenetics in the context of an embryo. As an embryo forms, numerous cells with different functions develop simultaneously. However, all these cells share the same genetic code. How is it that, say, an eye cell and a heart cell can have such vastly different functions while running on the same set of instructions? The answer lies in epigenetics and DNA. DNA contains the necessary information for a cell to function. It is then transcribed into RNA and translated into proteins, the basic component of all cells. Now suppose this segment of DNA contains the genetic code for a heart cell. Your eye cells don't need this information to function, so they communicate this through what's called an epigenetic marker. One example of an epigenetic marker is methylation. This is where a methyl group, which is a carbon-hydrogen compound, binds to a segment of DNA and essentially deactivates it. This in turn inhibits the production of the associated protein. Another example of an epigenetic marker is a histone. Histones are proteins that function as spools around which DNA can wind. Histones can boost DNA transcription by unwinding through a process called acetylation, but they can also inhibit gene expression by coiling more tightly through a process called deacetylation. Because of epigenetic markers, we thankfully don't have heart cells in our eyeballs, but the implications don't end there. While our genomes remain constant throughout our life, our epigenomes are continuously fluctuating. In fact, the word literally means above the genome. Therefore, our epigenomes can be influenced by factors such as smoking, diet, and medication. For example, cancer can be caused through the hypermethylation or excessive methylation of tumor-suppressing genes, or through the hypomethylation or undermethylation of oncogenes. Epigenetics affect not only physical characteristics, but also behavioral ones. For example, sisters and ant colonies share 75% of their DNA, yet they each fall into castes with distinct physical and behavioral traits, like warriors and foragers. It turns out that, just like with an eye cell and a heart cell, epigenetics play a role in dictating these differences. A study in January of 2016 found that by manipulating histone acetylation in ants, scientists were able to reprogram them to different castes. There's even evidence that these epigenetic changes are heritable. In 2013, scientists conditioned mice to associate a certain scent with pain from an electric shock. Surprisingly, the memory of this fear and the associated neural receptors were exhibited by the mice's offspring, even though the pups were never previously exposed to the scent. Scientists are still unsure of the exact mechanisms behind the inheritance of the epigenome, but studies like these imply that our decisions throughout life influence not only our epigenetic information, but also that of our children and grandchildren. Furthermore, scientists are beginning to consider epigenetic therapy to combat disease, especially ones such as cancer that involve epigenetic changes. For example, drugs that inhibit DNA methylation or histone deacetylation can potentially reactivate genes that have been silenced. In fact, Feridac, a histone deacetylation inhibitor, was approved by the FDA in 2015 to treat multiple myeloma, a form of blood cancer. Drugs like Feridac open windows of opportunity toward discovering new cures for cancer. At the very least, epigenetics will provide us a deeper understanding of our health and the role of our environment in regulating genes that make us more susceptible to disease.